Hi everyone, my name is Adriano Leuzzi from Retera Process Development, and today I will present you our GMP manufacturing process of adenoviral-based vaccine. So before to start, I'd like to spend a very few words regarding our mission. So Retera is a biotech company with a long-lasting expertise in viral vector technology. Working as a CDMO, we support our client for process development and GMP production up to phase 3 clinical trial and commercial material production. We also offer seroactivity for immune response monitoring to pathogen, vaccine, gene therapy, and immunotherapeutic drugs. Thanks to 8,500 square meter facility fully dedicated to the translational medicine, we are specialized to provide a tailored, timely, and effective solution to comply with the needs of viral vector market. Retera facility consists of a research and development lab for technology development and immune monitoring services, PD space for CGMP, scalable process development and improvement and analytical assay development for quality control tests, and a state-of-the-art CGMP facility with 1,700 square meters for production and release of clinical loads, assay qualification, and regulatory support to several kinds of viral vector, not only adenovirus, but also adeno-associated lentivirus, MVA, and herpes virus. So, why adenovirus are a good vaccine candidate? Adenoviruses have a long history as a DNA transfer vehicles in several medical applications as a vaccine agent for a wide range of infection disease. They are able to induce a strong and efficient humoral and cellular response by genetic engineering, firstly based on but not limited to the deletion of adenoviral genetic region encoding for protein crucial for viral replication. Adenoviruses are made replication defective and then saved. This is also combined with a high packaging capacity for foreign DNA as a gene of interest. Further advantages include high infectivity, high titer virus production, and compared to other vaccine technology as mRNA, they have also a low manufacturing cost and excellent product stability. So, in Retera, we have developed and characterized our proprietary cell bank derived from the original Frank Graham's HEC293 cell frozen in 1975 that are suitable for production of a clinical grade of non-replicating adenoviral vector that can be licensed as a vaccine. The adenovirus engineering strategy is well defined and characterized, lead them to be a very ideal platform for the emergent outbreaks and make this technology very flexible for a new potential vaccine production by introducing the transgene sequence in the adenoviral backbone. Combining this strategy with a fast-track manufacturing process, adenoviral platform can cover the global needs of a new vaccine and meets the regulatory requirements. In Retera, we have developed our proprietary vaccine platform technology based on a new gorilla adenovector produced in our packaging cell. Unlike the human adenoviruses, which are rapidly inactivated by the human immune system, those of chimpanzees are not as efficiently recognized by our immune system thanks to the low seroprevalence in the population, and they are able to induce a strong immune response both from T and B lymphocytes. So we offer to the client our proprietary production cell line derived from the Frank Graham cell that has been adapted to grow in suspension using serum-free synthetic medium. Our cells are engineered to complement E1 deletion and repress the transcription of the transgene carried by the vector, allowing for the propagation to high titers of a viral vector, ensuring high productivity and low frequency of vector genomic instability, making them suitable as a vaccine candidate. Now we can go deep into the vaccine manufacturing technology. As shown in this general flow chart, to produce a high amount of viral copy, adenovirus need to infect packaging cell and to replicate inside them. This step is made into Bireactor in which cells are seeded and grown until they reach a suitable cell density for virus infection. Once we observe a specific cytopathic effect, meaning that the virus reached our target productivity, the process proceeds with the next harvest and purification step, necessary to remove all process-related impurity up to final formulation in order to reach the final concentration and quality grade to meet the regulatory requirements. Our upstream process is based on high cell density to increase the cell biomass, ensuring high virus volumetric productivity, thanks to the implementation in our upstream process of a perfusion technology. As a consequence, some points need to be considered for a successful, reproducible, and scalable GMP process, especially the higher amount of contaminants in the bulk harvest, firstly whole cell DNA and whole cell protein, and the higher viral copy to purify. To solve this issue, we optimize our downstream process in order to obtain a product that is reproducible, analytical classification in terms of potency and contaminants, and that can ensure a very promising and good overall yield. So, our Adeno intensified commercial production process is composed by an upstream and downstream process. The process starts with our raised cell suspension adapted cells, thawing and amplification in shake flask. Then we proceed with the cell expansion in wave Bireactor to ensure enough cell density and enough biomass to perform the seeding in 200-liter single-use steel tank Bireactor. After 3 days of the seeding, we perform a perfusion process in order to remove the negative metabolites in the cell culture and sustain high cell viability and constant exponential cell growth. This can achieve the increased volumetric productivity after the infection performed into the production into the large-scale Bireactor 1,000, 2,000, up to full volume 3,000-liter commercial scale. After 3 hours post-infection, after 3 days post-infection, we perform the harvest step, and we start with the downstream process. The first step is cell lysis in order to collect whole viral particle, whole mature viral particle, and perform an host cell DNA precipitation to remove the DNA from host. So the next step is the cell debris elimination by that filtration in order to load our clarified lysate to the anion exchange membrane for fair chromatography step. So a DNA trimming step will be also developed and introduced into the process in order to further remove potential DNA fragments remaining by the process, by the upstream process. And then we continue with a polishing step by mixed mode chromatography to further remove any kind of process-related impurity, not only cell protein or cell DNA, but also endonuclease used in the downstream. Then the intermediate drug substance is ready for final formulation by TFF. As I said, more cell, more impurity. We developed our perfusion upstream process to increase cell volumetric productivity by ensuring and high cell viability and high cell available for the infection. But the first issue to be addressed is the removal of host cell DNA and host cell protein. In our downstream platform, we introduced an host cell DNA precipitation step based on a flocculating agent that enables the residual particles to link together in loose aggregates. These aggregates reproduce sediments while the virus remains in suspension and will be collected in the next clarification step, in which the resulting complexes of cell debris macromolecules complexed with a flocculating agent will be retained in the depth filter matrix while the virus passes in the flow-through. After the anion exchange membrane chromatography, that is the first purification step, able to reduce more than 2 logs of cell protein or cell DNA, we also introduced a new implementation in the process, an endonuclease digestion to remove osteoDNA fragments. As you can see by the flow chart, working on this smaller volume, you can reduce more the cost of goods, especially if you think in a final commercial scale. And this is a new implementation compared to the standard adenoviral process that usually perform the endonuclease digestion during the harvest step. And it makes real thanks also to the combination of osteoDNA precipitation and endonuclease digestion after chromatographic step. The best strategy in purification is to design an orthogonal process by using methods based on different principles. To further increase process-related impurity clearance, we include additional polishing step with a multimodal chromatography, essentially based on a dual functionality combining site separation and binding chromatography, in which smaller impurities are captured by the chromatographic matrix while the viral products are collected in the flow-through, and it is ready for the final tangential flow filtration, concentration, and the filtration into final formulation buffer. So, to conclude, in the landscape of the viral vector technology, over 150 adenoviruses have been characterized, and many of them are actually under study for vaccine development, mainly due to their characteristics as economical process-related cost, thermostability, packaging capacity for transgene expression, safety in terms of inability to integrate in the host genome, and capacity to induce a persistent and efficient immune response. From manufacturing perspective, the adenovirus production and purification process can be easily scaled to cover the global vaccine demand. And in Reitera, we have developed a robust and scalable manufacturing process based on implementation of perfusion for the upstream and on a downstream platform able to fulfill the regulatory requirement, ensure a very high final product overall yield that can prove as affordable vaccine solution up to the GMP commercial scale 3,000 liters, sorry. So thank you for your attention, and if you have some comments, we can discuss later in the next networking session, or you can also write or call me on Claudio, and you can also take a picture of the QR code. Thank you.